Chapter 10, Specific Defenses. Innate immunity is nonspecific. It's our defenses against any pathogen. Adaptive immunity is specific. It develops during an individual's lifetime. It is induced resistance to a specific pathogen. T and B cells develop from stem cells in the red bone marrow. So you'll see here that this is your red bone marrow. Both the T and the B cells start out here, and then eventually they both go in a different direction and then join up again at the lymphoid tissue. Humoral immunity. Humoral immunity involves antibodies produced by B cells. So it's due to antibodies. It's found in the blood, lymphatic system, and mucus. It is our defense against antigens and pathogens in the body's fluids. So it's our defense against bacteria, viruses, toxins, and transplanted foreign tissues. Cellular immunity involves the T cells. So it's due to the T cells that act against foreign tissue or organisms. It regulates macrophage release, and it's our defense against abnormal cells and pathogens inside cells. So it's our defense against bacteria and viruses inside of the phagocytes, fungi, protozoa, and transplanted tissue. So humoral immunity works on the outside of cells. Cellular immunity works on the inside of cells. Antigens. Antigens are a substance that causes the body to produce specific antibodies or sensitized T cells. Self versus non-self antigens. Self-antigens are the ones inside your body. Non-self are the ones from foreign sources. All cells and tissues contain antigens, but the ones inside your body don't stimulate an immune response. So all of our cells, so if we look at our red blood cells, that's how we know that we have different blood type. We have A antigens, right? So somebody that has A antigens is type A. Somebody that has B antigens is type B. Okay? So all of our cells and tissues contain antigens, but those that are inside of our body do not stimulate an immune response. Antigens are protein or polysaccharide, so they can be lipids and nucleic acids when combined with proteins or polysaccharides. They're found in the capsule, cell walls, flagella, fimbriae, and toxins of microbes. They can be pollen, egg whites, blood cell surface, and tissue surface. Antibodies interact with epitopes or antigenic determinants. This is where we determine the specificity. Okay? So the points on the surface of a pathogen that allow antibodies to bind are called antigenic determinants or epitopes. Okay, so they're the points on the surface of a pathogen that allow antibodies to bind. Each bacterial cell has many different epitopes. Let's take a look at an example. So here is a bacterial cell, and you'll see that we have like an oval shape, a square, then we have a triangular shape. Antibody A is specific for triangular shape because it has the lock and key sort of mechanism, you want to say, that's specific for this one. Would it be able to bind to the oval or the square? No, it wouldn't be able to. Okay, however, antibody type B, from what we see here, can bind to the oval, but we know antibody A will not be able to do so. So on the outside of bacteria, many different epitopes or antigenic determinants, okay, that will allow antibodies to bind. A hapten. A hapten is a partial antigen. It is a molecule too small to stimulate antibody formation by itself. However, when combined with a larger molecule, usually a serum protein, the hapten and its carrier function as an antigen and can stimulate an immune response. So haptens cannot stimulate an immune response by themselves, so they require a carrier molecule, such as a serum protein. Then the antigen and the carrier molecule come together and combine, and that together will cause an immune response. So an example of this is penicillin. Penicillin is not antigenic by itself, so when it's by itself it cannot stimulate an immune response. However, when penicillin combines with serum proteins, the resulting combined molecule initiates an immune response. So this is how we see individuals become allergic to penicillin, because it's binding to serum proteins. Antibodies are globular proteins called immunoglobulins made by B cells in response to an antigen. So B cells are the lymphocytes that produce antibodies. Antibodies are highly specific. They neutralize and destroy. So they do neutralization. So antibodies can bind to sites that would normally bind toxins from pathogens. This prevents the toxins from having any effect. Antigen binding sites bind to specific epitopes. The number of antigen binding sites determines the valence. So depending on how many binding sites you have, that is the number of valence sites that you have. So usually the most common one that we discuss is bivalent. So how many valence sites do we have? 2. Very good. So we'll have 2 sites. So bivalent antibody structure, a monomer structure. And that means that the monomer have 2 antigenic binding sites. Antibody structure. The antibody has 2 heavy and 2 light chains. So you can see this is the heavy chain, and then on the outside of the wings are your light chains. They are joined by disulfide links. So you can actually see the disulfide links here, joining the heavy and the light chains together. There are 2 different regions. The variable regions is found on both the heavy and light chains. The specificity of the antibody depends on the structure of the variable region. The antigen binding sites, if you see here, are on the free tips of the variable region. So here are the free tips of the variable region. If we blow this up, this is where we have that lock and key mechanism we were talking about. So that oval, right, or that triangle, this is where it's specific for that. The constant regions is found on both the heavy and the light chains. The constant regions of heavy chain forms the base of the antibody molecule. So here is your constant region. You have 5 different types of constant regions, and those 5 different types of constant region correlate to the 5 different types of antibodies. You also have what we call the Fc, or the stem region. So let's take a look at the 5 different classes of antibody. The first 3 that we're going to talk about are monomers. So they are monomers in structures. IgG, it's 80% of serum antibodies. It's found in the blood, the lymph, and the intestine. It is the most abundant antibody found in the blood. It has a half-life of 23 days. So when I say that it has a half-life of 23 days, that's half of its life. So what is its complete life? 46. Very good. So 46 days is how long that this antibody can last in the bloodstream. It can fix complement and cross the placenta. It enhances phagocytosis, neutralizes toxins and viruses, protects the fetus and the newborn. IgD is 0.2% of the serum antibodies. It's found in the blood, the lymph, and on the B cells. It has a half-life of 3 days, so that means that it's around for 6 days. Its serum function is unknown. On B cells, it's found to initiate the B cell response. IgE is 0.002% of serum antibodies. It has a half-life of 2 days. It's found on the mast cells, on basophils, and in the blood. It's involved in allergic reactions, lysis of parasitic worms, and it attracts IgG, complement, and phagocytic cells. IgM is a pentamer, and it's 5 to 10% of serum antibodies. It's found in the blood, the lymph, and on B cells. It has a half-life of 5 days. It fixes complement, and it stays in the blood, and it's very large. It agglutinates microbes. It is the first antibody produced in response to infection. It's involved in response to the ABO blood group antigens, so it's responsible for when we have incompatible blood transfusions. It enhances phagocytosis. It's very important in diagnosis because it's short-lived and it's only found at the beginning of infection, so at the initial infection. IgA is a dimer, and it makes up 10 to 15% of serum antibodies. It's found in secretions. Okay? So when I say secretions, I say tears, mucus, and saliva. So it's not very abundant in blood, but very abundant in secretions. So your tears, your mucus, and your saliva. It has a 6-day half-life, and it has complement fixation in which it activates the alternative pathway. It provides what we call a mucosal protection, and is our defense against respiratory infections and infant GI infection. Here ends Part A of Chapter 10.